Hello, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest of the beautiful USA. Today, as this first slide represents, is the 25th of November. That gives us exactly 1 month to get all of our Christmas presents not only purchased, but delivered, wrapped. This wrapping is really important. And then, of course, given to whoever is lucky enough to receive those gifts, right? So anytime people want to get started with that, I would recommend it. But today, we're going to talk about something different. It is the day before Thanksgiving, so at the end, I will remind you to have a fantastic Thanksgiving. But what we're going to talk about is the epigenetics of aging as it is associated with the immune response. And we've been leading up to this for some time. If you've been following along with the audio podcast on Authentic Biochemistry, you know that we've been detailing a great amount of information related to what are the genetic profiles of senescent cells, and then of the aging mammal, and particularly rodent models. But then most of our time we've been talking about human aging. We did a pretty careful analysis of neuroscience and neuropsychiatric conditions associated with aging in humans. That's a key feature of why we study it, really. We haven't talked a lot about sarcopenia, which is muscle senescence, but we've done a bit of it. We're going to do more of that because we're going to make the whole system, the human system, described as an aging towards death model of itself. And we're going to talk about the pharmacology of that, as well as the, of course, biochemistry, and then ultimately pathobiochemistry and pathophysiology of that process towards the later years in life. We have introduced many times the immune association with the aging response. And today we're going to do a more detailed analysis of that. And so that's one of the reasons I decided to flip this up on the screen and make a video of it rather than just do it on an audio. Plus, it gives me a chance to imagine the audience a little bit more clearly than I might when I'm just talking into a microphone. So here we go. I'll hide this bar down here. Okay, so this has a lot of writing, and that's because I've got a lot of information to get through. This paper comes to us very recently. It was published, as you see online, in July of this year. And what I'm doing is going to explain to you what this paper wants to get involved in its discussion with our own previous understanding of the aging process. And so this is what I've written in association with this paper to introduce it. Aging is both necessary and universal in humans. I say this because heterotrophic, multicellular organisms that multiply by sexual reproduction do not and cannot survive somatically. And since they do not reproduce offspring via simple cell division, nor do we generate whole new organ systems from simple mitotic division, or even division followed by some differentiation. So the human is essentially terminally differentiated, except during tissue repair, which does happen, thank God, often, but less so as we age. And then, of course, the other side of that coin is oncogenesis. And this, unfortunately, is one of the leading causes of death, particularly associated with old age. So this unidirectional sequence of lifespan is measured as a cellular and physical biochemical process. It's processive, and it's a senescence bounded by genomic foundations, of course. And it's tailored, though, immunologically via epigenetic modifications with those mechanisms in place, defines a temporal alteration of transcription factors, lipid and protein-based signaling trafficking patterns. This is both intra and extracellularly. Membrane dynamics, which has to do a lot with that signaling, but also with the biophysical alterations of the membrane surface, which we have gotten into in the past in authentic biochemistry, and we will again. And of course, the bioenergetic cost, which includes all the pathways to synthesize ATP. All of this ultimately determines an episodic and ultimately the final terminal cellular fate, right? Which then the whole organism also is associated with that. So physiological aging results from an incremental organ damage and failure, increased susceptibility to diseases. Now, right now, you can see why the immune system is brought into discussion. I hope that you know the immune system is not simply a defense mechanism for the body, but it's also the learning, tailoring mechanism for the body, particularly in the central nervous system. Hopefully, I've explained that or convinced you of that. But right now, we'll just say that susceptible to diseases and environmental stress, which doesn't necessarily require an etiologic agent, and the stochastic, that means random plenum of mutation, epimutation, and oxidative degradation, collectively understood as pathobiochemical morbidity. So all of that is where aging gets its ultimate presentations. Aging, and especially unhealthy temporal decline, is associated with obesity. We know this, unfortunately, over the last 50 years, increasingly important because morbid obesity has increased several-fold nationwide and worldwide. And we know that obesity is a primary risk factor in a lot of the human diseases. And many of these human diseases and their initial occurrence is accelerated in obese people as they age. Okay? So we know that obesity has something to do with at least components of the presentation of aging in humans. Not all, for sure, but many of them, especially those presentations that result in disease that then link to morbidity and to mortality. Okay. So major human diseases that chronically accelerate, can't read my own writing sometimes, during aging toward death include cardiovascular diseases. It's probably the major one, with cancer being right up there along with cardiovascular disease, depending on which sex you are. Neurodegeneration, Alzheimer's and Parkinson's disease, and of course a whole host of metabolic disorders like type 2 diabetes, metabolic syndrome, things like that. So there has been an increasing effort to uncover the role of epigenetic mechanisms. This is going to be the discussion this afternoon here in Authentic Biochemistry, in both a chronological aging and in just overall cellular senescence. Epigenetic changes are rendered via environmental stress, both internal and external. And as I've described numerous times, I guess I have to move my box here, in lecture, I knew that those words were there because I wrote them, involves lipid fluidity, histone modifications, DNA methylation, which is going to be an important topic today, small non-coding RNAs, chromatin remodeling, nucleosome positioning, and tissue-specific gene expression, all working through plastic and elastic molecular mechanisms. Okay. Protein and DNA methylation is the common covalent epigenetic biochemical process. It's an event that's been well described in the literature. So these processes, those processes are included along with histone lysine methylation. So you have DNA methylation, then you have histone lysine methylation, you have arginine methylation, you also have lysine acetylation, which we spent a lot of time on over the months, and serine phosphorylation. Most of those are covalent modifications which can be erased. In fact, all of them can, but not all of them do become erased, right? So that, of course, brings up whether or not something occurs necessarily or episodically, or it simply does occur or it does not occur. And a lot of that has to do with tissue specificity in aging. Okay. So those modifications alter the extent to which DNA is wrapped around histones, all those modifications, because they're chemical covalent modifications. So they're going to alter the chemistry and the physics of the macromolecules themselves, DNA and RNA and protein, right? And lipids as well. Wrapped around these systems and the availability of the genes within the DNA to be, once you unwind the chromatin, right, the complex, the core complex of histones with DNA, whether or not that DNA can be recombined, replicated, repaired, which is really important in aging, and of course transcribed into RNA. So there may be a unique role for DNA methylation in aging, and that's what this paper suggests. And so that's going to be the topic for today for us. So what are some of the mechanisms? Well, we know them because we are Authentic Biochemistry clients. So I'll go through them quickly. DNA methylation modification involves at least two different processes. The addition or removal of a methyl group on the 5th position of a cytosine residue, or the 6th position of adenine in DNA. DNA 5-methylcytosine, 5-MC, is the most prevalent, most popular of all those modifications in eukaryotic genomes. And it primarily occurs on what are known as CpG islands, or those positions in the nucleotide sequence that have a high register of repeating CpG, right, that dinucleotide. Repeating high levels of dinucleotide of CpG are often found in promoter regions, that is the portion of the gene that is not the coding region, but is exactly before the coding region, right? And the promoter region is actually where the RNA polymerase will eventually bind to and then make the transcript and then thus be involved, of course, in gene expression. So that's why it's so critical that there's a lot of the CpG there. If you methylate it, it does something physically and chemically to the DNA. So what does it do? The presence of a 5-methylcytosine, which of course is a bulky or relatively bulky hydrophobic group that discourages intramolecular rearrangements of the DNA, and of course because of that hydrogen bonding alterations, and it may inhibit transcription factors, including the all-important RNA polymerase II, from binding to the promoter region and therefore disallowing transcription because of that methyl group. DNA 6-adenine methylation, 6-MA, is far less studied, recently discovered, but it's still an epigenetic modification, of course, with its own enzymes. And it's found in the human genome, not just in animal models. And it has been demonstrated to affect, in fact, mammalian development. So this is a whole new area that's being looked at, but it's still within the same wheelhouse because it's methylation of nucleotides in DNA. So here's the process itself. So you got this chromatin here. That's what you're supposed to be imagining here. Here's the octet of histones that wrap around the DNA, right? The 256 base pairs of DNA that are wrapped around any of those nucleosomes. So this says here modulation of DNA methylation in mammals, including humans. You have 5-methylcytosine. The methylation site, of course, we already explained what that is. That's shown here. So you've got chromosomes, you've got DNA that's unwound. It's showing you now what it might look like. Here is the sequence of DNA. It's showing you methylating the cytosine residue. So that's what that looks like. On that same DNA, on another location, you can have this methylation of this adenine. That's what's shown here, and that's what this looks like. There's the methyl group. It's linked to a nitrogen atom here, right? It's linked to this methyl group is linked to a carbon atom, right? All right, so this is basically writing the methylation. Okay, it's writing it, but you can also have, once it's written, once you have a methyl group on the DNA, it's read by another protein. That is, it's detected by another protein. And by that, we mean that it is recognized, and that methyl group then will do something to how the rest of that chromatin is going to be handled by any of the enzymatic machinery that may be associated not just with transcription, but with recombination and repair, right? And with replication as well. So that methyl group has a lot to do with overall stability of the chromatin DNA once it's unwound from the histone. Particularly, that's when it's most active, right? During the S phase, for example, in the cell cycle. But the other thing to consider is you have to have a protein that recognizes that part of that DNA that is particularly methylated. And the more methylation, the more that protein is going to recognize those sites and then attract other proteins, making bridges and making complexes of bridges of polypeptides that will ultimately change the way genes are expressed. Now, the most common thing that happens is that adding a methyl group to a promoter region will inhibit transcription, but it's not always the case. And it's not just quantitative. The more methyl groups, because of the more methylated CpG or adenine methylation, doesn't necessarily mean there's overall less gene expression, but it's often the case because of that bulky methyl group. I mean, relatively bulky. Right? So you have writers, right? And those are methyltransferases. That's the enzyme that carries it out. Let's go back up here. Methylation marks are established by writers such as DNA methyltransferases and the N6 adenine methyltransferase isoform 1. There's actually more than one isoform of that enzyme. I think there are 3 now in human. And of course, those modifications are all then going to be looked at by readers, and that's what I showed you here. This is this green thing that's laid over this methyl group on this adenine residue on DNA. And again, those are these methyl-CpG or methyl-adenine binding domains, or just called MBDs here. And then you have erasers, and those are going to remove the methyl group and put the DNA back to where it was before that methylation occurred. Right? So this is really important. There's a couple of other enzymes that we're going to talk about. The TET proteins are really interesting. And also these ALKBH1s, and these are different kinds of protein erasers, which will affect the erasure of the methyl group, but it will do so in a more processive way because it's going to modify the DNA, and along the final end note of that modification, particularly with the TET proteins, you ultimately might get back to a non-methylated residue where you once had one, if it happens before replication. Replication was going to, of course, change the nucleotide sequence when there's a methyl group there. And we'll talk about that as well. Okay, so you have writers, you have readers, and you have erasers, okay, or any epigenetic phenomena. And the one in particular we're talking about is methylation, right? And those 2 methylations, right? You have a 6-MA or you have a 5-MC. Okay. All right, now more about the machinery. This is kind of interesting stuff, and a lot of it is pretty new. Even though epigenetics has been, the science of epigenetics, the subscience of biochemistry known as epigenetics. These are all biochemical sciences, by the way, not genetic, because to study it, you have to use biochemistry, not just plain molecular genetics, because there's a lot of enzymology, and you have to know substrate product interactions and relationships. You have to know how to put this into test tubes, put this in the cell lines, and that's more of the realm of a biochemist. But those of you that watch and listen to Authentic Biochemistry are probably well aware of that, because some of you are biochemists or you want to become biochemists. And I recommend that if you do want to become a biochemist, you are not hurting yourself by listening to Authentic Biochemistry. Okay. All right, so DNA methylation, enzymatic machinery. You got DNA methylation, 5-MC and 6-MA, and they each require a unique enzymology. The 5-methylcytosine methylation is the result of DNA methyltransferases we just looked at, and those include DNMT1, DNMT3A and 3B. Don't worry about DNMT2. It does exist, but it does something different than what we're talking about here. It does something besides epigenetics, actually. Now, DNMT1 is primarily responsible for the maintenance of methylation across mitosis, so that's known as maintenance methylation, right? So indeed, methylation can be maintained even after cell division. Okay? While the DNMT3A and 3B work in a de novo methylation process, like adding methyl groups to either cytosine or adenine residues in the nascent DNA sometime before mitosis occurs to locations that were not otherwise methylated. And then that DNMT1 is involved in methylating on a pattern-specific basis, that sequence of DNA that's always methylated. And that happens a lot when you have silencing of genes. For example, when you have multiple copies, or if you have only 2 copies inherited, for example, from the mother and father, one of the ways to silence one of those copies of genes is to add a methyl group, and that's the business end of DNMT1. Okay, so 5-methylcytosine methylation can be oxidized. This is important here, and changes into 5-hydroxymethylcytosine by this protein called 1011 translocation, or TET, okay, enzyme. And they contain TET1, TET2, and TET3 as different isoforms. Now, this is a curious stuff. Let's take a look at this. So these are the pathways of cytosine demethylation by 1011 translocation. Okay, you have adenosine deaminase that's involved. So you're going to already have this initial reaction here, right? You're going to make 5-hydroxymethylcytosine, okay, which is this product. So you've got cytosine. You do the methyltransferase. It doesn't matter if it's 1 or 3A or 3B. You've got now this methyl group on here, 5-methylcytosine. This is a degradation of that methylated nucleotide. So it's removal of the epigenetic marker, if you want to look at it that way. So this 5-hydroxymethylcytosine will then be reacted upon by the enzyme adenosine deaminase, and it will synthesize 5-HMC, okay? And so that is 5-hydroxymethylcytosine from hydroxymethylcytosine. And so this will then, or uracil, excuse me, so 5-XMU, this is the uracil because you've carried out the deaminase, I'm sorry. So now you've got 5-HMC uracil, and that's going to ultimately be greeted by the next enzyme, final degradation pathway, enzyme TDG, which is thymine DNA glycosylase, going to remove sugar, ribose sugar. Then you're going to go through base excision repair, which I went through very recently as one of the ways to repair DNA. And base excision repair can build back up cytosine directly in the DNA sequence. Okay, now that's one pathway. The other is more reaction with TET. Okay, it's going to fake 5-FC, that's 5-formyl, that's formal group cytosine, and that will directly be deglycosylated. Okay, and when that happens, it goes through base excision repair, and you can resynthesize cytosine. The final reaction with the same protein TET, okay, will make this last one here. Trying to remember what. Okay, here it is. It's 5-carboxyl cytosine. That's what that is there, of course. And then ultimately go through this DNA glycosylase, go back into base excision repair and make, again, bring it back to cytosine, which means that that is an erasure of that initial methylation. Okay? So you go through the 5-HMC to 5-HM uracil, 5-formyl cytosine directly into the base excision repair, or the 5-carboxy or carboxyl cytosine pathway, then into the glycosylase. All of these end products of these TET reactions can lead back to, because of base excision repair, cytosine. Okay? All right. So that's the new potential fate of methyl cytosine residues. So, now, there's more to it than that. DNA methyltransferases can also act in reverse, okay, because it's just a transferase reaction. So the equilibrium, the amount of energy that's required to go in one direction or another, usually has to do with substrate concentration, right? And whatever cofactors may be associated with these, and there are some. So DNA methyltransferases can act in reverse, and therefore they can demethylate, therefore they can erase as well as write. 6-methyladenine methylation is generated with this N6-adenine methyltransferase 1 and this enzyme called ALKBH1, but the level of 6-MA in cells has not been completely confirmed to exist after mitosis. Okay, so it shows up, and we do see it, but we don't know what its real role is, although it does seem if we don't get 6-methyladenine production and also ultimately removal, we don't get development in some mammalian models. Okay? So there are promoters and enhancers at typical sites, of course, for DNA methylation, and that's where the methyl-CpG-binding proteins are going to be found. So if you're looking for them, then you're going to try to find CpG islands, and you're going to find MBDs, and then you can isolate those proteins and analyze them for substrate specificity and also for function. Here's a paper that came out a long time ago. I mean, not a long time ago scientifically, or even really chronologically, but 6 years ago, okay? Why am I going to this paper? Because I want you to have more information about the TET, okay? This is where it was first discussed. 1011 translocation, or TET family genes, initially found as a chromosomal translocation between chromosome 10, right, and chromosome 11. That's why it's called 1011 translocation. Now, those of you that are medical doctors are aware of the fact that when you get chromosomal translocations, many of those diseases end up as leukemias or blood-borne cancers, okay? Indeed, this is one. So this 1011 translocation phenomenon was discovered in leukemia. It turned out to be a key enzyme once they studied the protein, okay? So they isolated it, probably genetic, by a genetic screen, so I'll give the geneticists credit there. But then the biochemist got to work and gave you all the detail that I just showed you. So what these proteins do is hydroxylate, as you just saw, to 5-methylcytosine, to 5-hydroxymethylcytosine, and then converting it to an unmodified cytosine via the basic system repair pathway, right? And using the AIM enzyme to the DNMTs. You're going through uracil, which is only one of the pathways. Somatic mutations of TET2, one of the isoforms of the protein, were reported in a variety of hematological malignancies, right? Blood-borne cancers, such as leukemia, which I've already mentioned, myelodysplastic syndrome, or malignant lymphoma. And they all suggest a critical role for TET2 in hematopoiesis, okay, obviously. So the TET-mediated cytosine demethylation pathway, which you just saw, is linked to a mutation of, interestingly enough, isocitrate dehydrogenase. So there's isocitrate dehydrogenase 1 and 2 are found in the mutations in those two isoforms of that enzyme are found in hematological malignancies. The mutation inhibits, when you get the mutation in that enzyme, it's an isocitrate dehydrogenase enzyme, which I know you've encountered in the TCA cycle, right? Right? Here it's a cytosolic enzyme, though. But because there's cytosolic isomers of IDH, we talk about them in metabolism all the time. Cytosolic form, mutation in those proteins is going to cause a product form which is going to inhibit TET function. And it's going to do through that metabolite, it's known as an oncometabolite because it's going to cause these cancers, unfortunately. It's 2-hydroxyglutarate, 2-HG. So mutant TET2 results in the expansion of multipotent as well as myeloid progenitors, and that can lead to the accumulation of pre-malignant clones. This is another way of getting malignancies, by getting these mutations, you're saying. In addition to cytosine demethylation, TET's also involved in chromatin modifications other than that, as you might guess, and other cellular processes through the interaction of oxygen-linked beta-N-acetylglucosamine transferase, or OGT, which is an important enzyme actually in lipid biosynthesis. So these enzymes play a role in lipid metabolism. They surely do. Functional TET2 is a critical regulator of also hematopoietic stem cell homeostasis. Okay. Right. So this has to do with, of course, with development and differentiation. So this is showing you this mutation here. This comes from a paper published in Cancer Science, June 2014, as I said. Inhibition of the 1011 translocation mediated DNA demethylation processes by a mutant ICD, IDH, excuse me, either 1 or 2, and you make 2-hydroxyglutarate. So you got isocitrate, and normally you make alpha-ketoglutarate. But when you have a mutant form of isocitrate dehydrogenase, okay, you don't make that, you make 2-hydroxyglutarate, right? So this is the mutant enzyme down here. Right? I don't really like this slide because it looks like you make, you have to go through multiple pathways. That's not really what happens here. But IDH2 will make 2HG. 2HD will then will block histone demethylases. It will block histone demethylases. It will also block TET. So it totally prevents or removes that epigenetic signature, right? So alpha-ketoglutarate actually helps activate histone demethylases. But you don't make alpha-ketoglutarate, you make 2HG, and that blocks the reaction. 2HG also blocks the TET offensive pathway, taking 5-methylcytosine and making cytosine. The reaction is what you just saw, okay? So you get a mutant enzyme in basically carboxylic acid metabolism, which stops at two levels the activity of enzymes that are involved in removing methyl groups. So that means the methyl groups are retained, and because of that, the mutations result in some instances with hematological oncogenesis. So genome-wide DNA methylation levels are associated with progressive age. So this is interesting, okay? Higher levels of methylation. And it's more prominent in specific regions of the genome. So that suggests you're picking up all these methylation patterns. They don't go away. They don't get demethylated as you age. And in fact, in certain regions of the genome, and that tells you that that could have a pathobiochemical response, right? An outcome. So it's certain because it's involved with aging and morbidity, right? And ultimately mortality. So in certain circumstances, DNA methylation biomarkers have been proposed. Aging is associated with cell senescence, of course. Dysregulated nutrition, right, which is also really important. Dysregulated nutrient sensing associated with dysregulated nutrition. That has to do with adipokine and endocrine and paracrine hormone metabolism. Okay. Stem cell exhaustion, which is also critical to getting ultimately senescence. And using biomarkers like this protein P16-INK4A, or the CRP protein, or creatinine, fasting glucose, telomere length, these are all biochemical markers that can be looked at for aging, right? And of course, the reactive oxygen, which is going to cause the oxygenation and ultimately the oxidation of DNA, RNA, protein, and lipid. These now we're talking about getting these epigenetic signatures, and that has to do with this TET protein that we just talked about. Now, this is all described in a paper in Genome Biology 2019, so it's a relatively recent paper. Now, I'm going to remind you what P16-INK4A does, okay, because it's apparently a biomarker for aging. Now, curiously enough, we've talked about this a couple of months ago. So let's go back and discuss where we were. So just to remind you, because I know that as soon as I do, you'll remember this immediately, or you'll go back and watch that lecture. Paper that was published just this year, 2020, in BMC Cancer. Germ cell testicular cancer mainly affects young males between 15 and 35. Really sad disease. It's estimated that there were 71,000 new cases and over 9,500 deaths worldwide in 2018 when the numbers, last numbers were first compiled. So it's a deadly disease. It is the most curable solid neoplasm, however, and even if it presents in advanced stages, it can be cured with current chemotherapy. So it's not a cancer that can escape even basic chemotherapeutic means to destroy the cancer and give the person back their years of life. 90% of the patients can be cured with current chemotherapy, but 90% of them with good risk disease will be cured. The intermediate risk group has a 5-year survival rate of approximately 80%, and patients with poor risk disease, unfortunately, 5-year survival is only about 50%. So these TCS patients struggle, though, because they're young. Remember, they're 15 to 35. This means many of them before reproductive age. Chemotherapy can destroy reproductive capability, right? But why? Because it kills nascent cell synthesis, right? Because of that, spermatogenesis is affected, right, in males. So TCS patients struggle with the long-term toxicity of oncological treatments, such as normal chemotherapy. Side effects include increased incidence of secondary malignancies, because they're young and they're still differentiating and developing. Hypogonadism, unfortunately, of course. Pulmonary toxicity, nephrotoxicity, neurotoxicity, augmented mortality from circulatory diseases, and increased risk of infections. So TCS have a higher risk of dying from all those other non-cancerous causes, except for the first one, which is a secondary malignancy. It's really not a good thing. So clinical manifestations associated with immunosenescence in the elderly include, of course, a decreased ability to control infection. Poor response to vaccination is also common in the elderly, and an increment in the risk of developing cancer. Several immunological alterations have been described with aging, including changes in T and B lymphocytes, which we've talked about recently, subpopulations of those two families, plus natural killer cells. So the B lymphocyte compartment also shows a decrease in naive B cells with a reciprocal increase in memory B cells, which have their own potential pathology. Aging is also characterized by a decrease in circulating antibody levels. These would be antibodies already raised against antigens through the lifespan, such as ones encountered with immunization or with natural presentation of antigens from potential pathogens in circulation or in tissue beds. At the functional level, natural killer cells exhibit decreased cytotoxicity, so they're not able to act as good cells to remove host cells that are diseased. And particularly natural killer T cells also have reduced migratory capacity, right? The T lymphocytes, right? Natural killer T lymphocytes. So here comes this P16 INK4A protein, which we just mentioned to you. This is a P16 INK4A is a cell cycle regulating protein, and it's an inhibitor of a cyclin-dependent kinases 4 and 6, CDKs 4 and 6. It plays an important role in cellular aging, and the premature senescence has been recognized as an aging biomarker. Okay? So P16 INK4A expression in peripheral blood T cells has been described to sharply increase with age. So remember, we want to look at what these proteins, just because you see a protein as a good biomarker doesn't mean that protein has an effect on the process you're looking at. Well, you don't know until you study it. But it does contribute to age-induced functional decline of certain self-renewing compartments. Like what? Like lymph glands. Okay. Or like what? Sertoli cells. Okay. So here's a paper published in 1999, Cancer Genetics and Cytogenetics. You can see the reference there. Scrotal cancer is the first described occupational cancer that is in the literature, actually. It's where the human papillomavirus, oncogenic type 16 and 18, are reported as the causative agents in the pathogenesis of those cancers. You've got E5, E6, and E7 proteins expressed by this virus. Virus type 16 is the biovar of that virus. It's the most virulent, and so it has a particular name, virus type 16. It affects those proteins, affects cell cycle at the G1 checkpoint. So TP53, P16 INK4A, and P15 INK4B are all reported as the transcription factors that regulate the cell cycle on a same pathway. They work coordinatedly. mutation pattern of those 3 proteins within the genes, that is, and the homo or hemizygous deletion patterns, at least of the INK4A proteins, is associated with scrotal carcinoma. So obviously, these proteins have the potential to describe oncogenesis, right? Survival rate of the 2 human papillomavirus 16 positive patients who had a TP53 mutation or one of those INK pathway mutations, if you have a homozygous deletion or both, was lower than what you require for human papillomavirus 16 negative cases who had no TP53, P16-INK4 and P16-4B mutations are found. So the molecular alterations of TP53 and the INK genes can be useful, as I've said, as prognostic markers in scrotal cancer. This is when this was first described, only, 21 years ago. But still, that's ancient history in scientific literature. Now, jump way up to 2017 now. It's only 3 years ago. Senescent cells accumulate in many vertebrate tissues with age and contribute to age-specific pathologies, presumably through the secretion of factors contributing to senescence-associated secretory phenotypes, SASP, right? And we've talked a great deal about this. It's published in Nature Medicine, right? So removal of these senescent cells may be useful, right? Because the pathologies associated with those senescent cells seem to limit a healthy lifespan. So aging and trauma are risk factors for the development of osteoarthritis. We've known that for a long time. Of course, it's a chronic disease characterized by degeneration of articular cartilage, leading to pain and, of course, physical disability. Senescent chondrocytes are found in cartilage tissue, isolated from patients undergoing joint replacement surgery, okay? Yet their role in disease pathogenesis is not well known. To test the idea that the senescent cells, magic glasses back on here, might play a causative role in osteoarthritis, they used a P16-3MR transgenic mouse. That harbors this P16-INK4A, its other name, of course, is cell cycle D or CD kinase 2A. That promoter, which drives the expression of a fusion protein containing a reporter gene called the renylluciferase, and the monomeric red fluorescent proteins. That's 2 different, it has a 2-component system to act as a bioluminescent marker, basically. So it hooked up to the promoter for P16-4A. Okay. You get a truncated form for herpes simplex virus 1 thymidine kinase gene that's going to be used for selection of this transformation of this mouse line. Okay. That's using a negative selective marker called HSV-TK, which I said when I first talked to you a while back that the authors used this as well. Your presenter, well, it was also an author, used that same gene system for negative selection. So the mouse strain allowed us to selectively follow and remove the senescent cells after anterior cruciate ligament transection, or ACLT. Okay, so let's go through this now. When the we in here is not me, the we in here are the authors of this paper, of course. All right. They found that senescent cells accumulated in the articular cartilage and the synovium after this surgery. And there was selective elimination of those cells. When they did that, attenuated the development of the post-traumatic osteoarthritis. Also reduced pain, which is really good, and increased cartilage development, which is further good. Intra-articular injection of a senolytic molecule that selectively kills these cells, these SNCs, validated the results in transgenic, which I just told you, and the non-transgenic and aged mice, which also develop osteoarthritis, of course. Selective removal of the senescent cells from in vitro cultures of chondrocytes, isolated from patients that had OA, undergoing total knee replacement, decreases finally the expression of senescent and inflammatory markers, while also increasing expression of cartilage tissue extracellular matrix proteins, which in this case are promoted. So they wanted to test whether senescent cells contributed to the pathogenesis. So do the senescent cells develop after articular joint injury in mice by following the expression of that protein, which is, again, this cyclin-dependent kinase inhibitor 2A. And they want to know if that could be a frequently used biomarker now for these senescent cells. So they induced a post-traumatic osteoarthritis by this surgery. The surgery induced it in a P16-3MR transgenic mouse. Now that's the mouse that has this whole P16-INK4A promoter hooked up to that bioluminescent biomarker as controlled by the homologous recombination event that they're looking for here via negative selection. And also negative selection allows them to throw out any of the transgenics, just to have random integrations, right? So that's how this is done, right? By negative selection. You add this ganciclovir to the medium, and you kill all the cells that are not transgenic because they're expressing the tyrosine kinase, so you can get rid of those cells. You can eliminate them, right? So in negative selection, in homologous recombination experiments, you use that to get rid of anything that is outside the region of insertion, which is a homologous recombinant. That's how negative selection works. Here they're using the thymidine kinase chain to kill these cells after they've altered the expression of the bioluminescent biomarker. So there's a lot of different ways of getting around doing this research. And I talked about this in the past. So they induce the arthritis by the surgery in this population of transgenic mice, which already had this all set up. And that setup is that you have P16-INK4A positive senescent cells, and they could be non-invasively monitored. Why are they non-invasively monitored? How do you do this? By looking at the biomarker, right? So you look at bioluminescence. It's a nice way of not destroying the cells in the process of looking at which cells are expressing this biomarker, which also now have OA, right? So luminescence in the articular joint region of those mice, in which that P16-INK4A positive senescent cell was monitored. What happens there is you get increased after ACLT surgery. That's about 2 weeks after that, you get this luminescence building up. And then there's a decreasing thereafter to a very low steady state. So it seems that whatever pathology is occurring right after surgery. So this is good information for them to obtain. And that remained above the background, though, and it was coincident with mRNA levels of this CDKN2A, this kinase inhibitor that we're talking about. And then it decreased with kinetics similar to those of the luminescence. So they're following the luminescence, but they're also then screening, that's not selection, that's screening for the activity of that P16-INK protein, right? So the RNA of CDKNA1A and I16 or 11 or 116 encode a SAS marker, which was interleukin 6. Okay. They were also higher in vehicle treated, so they didn't do anything else except just look for whether or not you had SAS activity within the background of this transgenic population. So vehicle treated ACLT mice, then sham operated controls a full month after, 28 days after the surgery. So that further, all of that corroborates the development of CNS or senescent cells being involved in the injured joint, which then leads to the osteoarthritis. So they tell you that osteoarthritic disease and cartilage degeneration after the surgery were confirmed histologically, and of course they looked at pain assessment. So it is possible, okay, so let's move out from that experiment, that testicular cancer survivors are immunosenescent because they have the same biomarkers and the same associated diseases, some of them, that you get with aging. The whole reason we went into that OA experiment is to show you that P16 INK4A protein and 4B protein, which, remember, are regulating cell cycle because they're kinase inhibitors, right, are also involved in the induction of osteoarthritis after joint surgery. Right? And all of that is linked, those morbidities are linked to aging and you pick up on that P16-INK4A biomarker in these young men, right, who have gone through chemotherapy, but because they had cancer, right, testicular cancer. So you get the idea how all this is functioning together. Those are 3 or 4 different avenues of research looking at what is related to aging. And so this young population of young men that got cancer and then were given chemotherapy and then have advanced aging subsequent to that, they want to find the lineage. And the lineage, they look for particular biomarkers. Doesn't mean the biomarkers are causing the aging or related to the age-specific disease. It only means it's a marker. They could be related to the pathophysiology or pathobiochemistry, but that has to be determined. So, data from a paper published in BMC Cancer in 2020 demonstrated there was increased expression of P16-INK4A in CD3 lymphocytes, okay? And that protein is associated with immunosenescence in the elderly. Now that's all very interesting. Now you've got the immune system because you're getting a buildup of this protein in these CD3 positive lymphocytes, right? Which are ultimately going to differentiate into full-blown T cells. And that's all in association with immunosenescence. So the next question you have to ask, though, when you got that far, does this offer conclusion? Can you decide that that's something we can go forward with via induction and then make a new hypothetical deduction from other data? Does it offer conclusion that testicular cancer survivors treated with conventional chemotherapy indeed acquire an early onset immunosenescence because they seem to senesce quickly? Or merely that the biomarker is there? And I already gave you that argument. That could well be the case, right? Even if there are pathobiochemical signatures in the population that cohere with elderly immune sundowning, is this translated to functional phenotype? Now, there's another whole interesting component which we've been dealing with, chronological clock, right? Well, chemotherapy-treated testicular cancer survivors do present reduced naive T cells, but with a concomitant increase in TMEMs. And that's compared to healthy age-matched control adults. So, or people in their age group, young adults. All right. So let's step back from that. You're starting to get reduced naive T cells, increase in T memory cells. Remember one of my previous lectures, I told you that one of the hallmarks of the aging population is a lack of naive CD4 positive, CD8 positive T lymphocytes, either in the periphery or coming from the lymph, or coming from the thymus. And that because of that, right, you have less of a robust response because you have less naive T cells to respond to a new invading pathogen, for example, or some xenobiotic event. You have T memory cells, which, remember, when they are triggered errantly, they can cause a hyperimmune response and an increase in inflammation. Chronic inflammation can lead to things like tissue death, of course, neurodegeneration, and what? Can also trigger cancer, right? So all these come together because those are the major diseases we just talked about that are found in the elderly. So you get out where all this comes together, right? All right. So, indeed, the ratio decrease in T naive over memory cells is common in the elderly. I kind of already let that slip out, right? There are healthy matched controls. Are those a good control, though? Are healthy age-matched controls a good control? Because they didn't have the cancer and they didn't get the chemotherapy. There's 2 things missing there. So how can you separate the chemo just from the testicular cancer patients? So it's critical to evaluate the mechanism for premature immunosenescence in that population. One of the problems is you could, of course, do a study with testicular cancer patients prior to pharmacotherapy, including chemotherapy, or maybe programmed death ligand is sometimes being used now for that cancer. And so it's just a highly treatable cancer just with chemotherapy. But how long are you going to have them to study them before you want to give them treatment, right? It's unethical. So it's difficult to work in that population, okay? Now, as humans age, there is an increase in CD57 positive, terminally differentiated senescent cells. And they have a reduced proliferative capacity and they have altered functional properties. In fact, these testicular cancer survivor patients, which had chemotherapy, had elevated CD8 positive, CD45 retinoic acid positive, CD57 positive cells as composed to controls. So 41.6% versus 24.7%, with a pretty way good p-value, 0.015. So it looks like statistically significant, and that you can separate those two lineages between being TCS patients and being controls. And remember, those are going to be healthy controls. So TCS exhibited a trend towards an increment in CD20, minus cells, right? And a group of pro-inflammatory cells that increase gradually with age and may account for up to 50% of the geriatric population, right? So I told you some of those cells, they're already pro-inflammatory. They're stationed that way. They cured that way into terminal differentiation. And those are the ones that can be turned on and make a hyperimmune response, and therefore chronic or a high level of inflammation, which can lead to all that tissue damage. So the percentage of CD28 cells in the control group was 1.5%, and that's well within the respectable expected limit for the young adults. So at least these CD8 negative cells don't seem to be playing a major role here, right? Unlike the increase in the CD8 positive, CD8 minus Tregs, these are the Tregs now, in TCS patients, B lymphocytes showed no significant difference between case controls and naive cells. Okay, so recall now, we're almost at the point of a junction here with P16 INK4A. Recall that protein is a cyclin-dependent kinase inhibitor during cell cycle. And it's also been well regarded as an aging biomarker because it's easy to catch it in the serum. That renders the senescence growth arrest irreversible because it blocks what? Cell cycle progression. Yeah. Right. So the CDKN2A gene expression is also increased in women, interestingly, with breast cancer exposed to chemotherapy. But go back and think about this. The CDKN2A gene, okay, which is, of course, the P16 INK4A protein, will render the senescence growth arrest irreversible. So you have senescence growth arrest, right? And you make that arrest irreversible. So what's going to happen? You're going to get senescent growth, right? That's what it means. Okay. So CDKN2A expression in all CD3 positive T lymphocytes, right, they're called PPTLs, is a good biomarker of senescence, right, since there is about a 16-fold change over a human lifespan. Right. So the combined effect of this gene expression inhibiting the proliferation of aging T cell populations and the loss of the CD28 lineage serves to enhance the immunosenescent phenotype. Okay? Now that's been explained and may be made florid in healthy elderly as well as chemotherapy treated younger male adults after testicular cancer. Okay? Ultimately, then, what we can say is this T cell phenotype confers an increased susceptibility to infection, enhances the potential for chronic infection and chronic inflammation, and it potentiates mechanisms for autoimmune diseases such as MS and lupus and arthritis. It can also be associated with tumorigenesis, cardiovascular disease, and of course metabolic disorders, which are rampant also in the elderly, particularly obese. All those are linked to later in life morbidities associated closely with human mortality. Right? All right. So that BMC paper offered a perspective then on the differences of lymphocyte phenotype and the expression of that protein in a healthy cohort versus a compromised TCS population. Remember, that's testicular cancer survivors. Now, ironically, Frank type 2 diabetes and hypogonadism alter the P6-INK4A expression in non-testicular cancer populations. So it could be difficult to separate the effects of that expression of that protein as necessary, possible, or simply coincidental with testicular cancer or with testicular cancer survival. Now, I first pondered that at the beginning of critiquing the manuscript, remember, way back about 15 minutes ago. So it's a cross-distributional study, so cohorts were not separated out a priori. So you couldn't eliminate with experimental design, you couldn't eliminate any kind of experimental design bias, right? Which could contribute to the whole variation that we looked at, even looking at the effector analyses. So it's therefore more difficult from that caveat, the understanding of the concept of that caveat, more difficult to determine which, if any, of the variables contributed to the variation in the data. So statisticians love to tell you this at the end of an experiment, but they're also pointing at something that's biological, right? Because the statistics are only as good as what the biology is and nothing more. And often statistics are used to corrupt what the biology is saying. And that's often because there's some agenda involved in the research. And sometimes there simply is. All right, now, cross-sectional studies like this are still important, okay, even though you haven't been able to develop cohorts, because they produce data that is more natural to the human population, right? You're not segregating the population according to other morbidities or other pharmacotherapy that's involved, or a distribution based on race or some other composite that you may or may not think has anything to do with the disease, right? So cross sections, you don't do any of that. You just get whatever the population is, right? So it's more natural to the human population when you do cross-sectional studies. For example, in this case, the correlation of that gene, CDKN2A, increase in the CD28 minus phenotype could suggest the two systems are related both at the level of gene expression and at the level of antigen presentation interactions because of the effect on the T lymphocyte population, you see? Now, this is how you can theorize with a good understanding of the biochemical system, right? This is what you do when you read these papers. You have to understand what it's telling you from what it even suggests it's telling you, right? So you have to go 1 or 2 octaves above it, and you have to deploy logic, and you have to employ the square of opposition to analyze this data and to understand it within the population of all the other research papers, but also within the population that you understand how your mind is working when you're analyzing it, right? So that antigen-presenting cell interactions with activated T cells is important. And also we have to think about epigenetic factors, which is how I started this lecture, or factors as yet not isolated, not dissolated. It's an interesting word there, from the population. The spelling sometimes on these slides is rather unfortunate. Anyways, to be sure, testicular cancer survivors were analyzed at different time points after treatment. There's no evidence, really, that P16-INK4A variation simply with a short time interval is going to change. Okay? We just don't know. Recall, though, that the testicular cancer survivors are cancer plus chemotherapy, right? So the effect of chemotherapy versus cancer by itself remains to be defined. You can't do it, at least not with this experiment. There are no differences in CDKN2A expression between patients that received 3 or 4 cycles of BEP. Now, that's a drug that's used, right? That's the drug, that's the chemotherapeutic. Or patients that received 1 or 2 regimens. So it's not titratable. This doesn't look like whatever's going on here with this P16-INK and whether or not it comes up because of the chemotherapy is titratable, which may suggest that the cancer itself, sensu stricto, or the cancer in combination with the chemotherapy, but not quantitatively, only qualitatively, show up as a possible linkage of P16-INK4A comorbidity with senescence. and oncogenesis in the elderly. Take-home message from that paper could be, maybe, that immunosenescent phenotype in the elderly, while associated with an increased rate of infection and diminished effect of vaccines and increased cancer susceptibility, may be promoted by cancer, okay, and or chemotherapy. So immunosenescence may be promoted by cancer or by the therapy used to treat cancer, to cure the cancer itself, right? Now, both will contribute to immune compromise, of course, and potential for gene mutations and epigenetic for them. So the whole map is opened up. The authors are, after all, clinicians, right? So they stress that their work reinforces the importance of considering surveillance before or over chemotherapy or even radiation therapy in the very early stages of testicular cancer. And of course, that's a very important point. That's why they like these biomarkers. They just kind of stumbled on this immunosenescence, but it's also a clever thing that they found, right? So the authors conclude that TCS, remember, testicular cancer survivors, to chemotherapy presented multiple alterations in lymphocyte subpopulations, which is interesting, an increased expression of that p16-INK4A, which, remember, is an inhibitor of cell cycle, so it will arrest terminal differentiation of senescent cells, making them last longer. And all those alterations are akin to elderly immunosenescence, because it's the cells that are senescent that we're describing here are, of course, of the T cell lineage. Okay. So back to this paper, Genome Biology 2019. A DNA methylation clock, then, that they're discussing in this paper could be an estimator built from epigenetic DNA methylation markers. Okay, so the reason we went through this whole discussion of testicular cancer, which comes from the p16-INK14A, is so you see where the other literature fits into place. Once you start getting more and more literature together, you can generate a hypothetical deduction. And that deduction then will result in some kind of experimental design to test the null hypothesis, to generate data that can be converted after the study of the data into potential evidence, and the verification of evidence against the bulwark of all the other papers published, which is what we were just doing here. So now I'm coming back to the paper we started with, Genome Biology. They say a DNA methylation clock is an estimator built from epigenetic DNA methylation markers, which are strongly correlated with chronological age or time, might be able to accurately quantify age-related phenotype or an outcome to a disease. Okay. DNA methylation clocks involve computational, penalized regression trained against chronological age. So this is how they use this methylation clock to generate algorithms to study age against this methylation, which they think is a good marker for aging. And then they look at readout for disease. I think it's clever. So people that study genome, that's what they do. Some do. And they want to identify an informative, sparse, predictive set then. They want to find a nested population of CpGs that are methylated. So not only do they have the methylation clock, which is quantitative, they want to know which CpGs are methylated. That's qualitative. So the two come together. Then you get real, what? The ability to do three-dimensional biology, which means you do it over time, which is exactly why the elderly population is so good to use in this kind of study, besides the fact that the elderly are the ones you're studying. So the age-related phenotype or outcome may be disease. So you can look at this all from the DNA methylation clock associated when disease come up, which diseases come up. More methylation relate to one kind of disease over another, one mutation over another, one epimutation over another, one epigenetic phenomenon over another, not just methylation. You get the point. Mortality could be another endpoint. Do these methylation clocks predict when, or not when exactly, but whether or not a person will die earlier rather than later, or even mitotic age? And the mitotic age is the total number of lifetime cell divisions of a given tissue cellular population, right? The older the mitotic age, the more you reach senescence, right? And reaching senescence then is the natural phenomenon for all of us. It's aging. But reaching it at a critical point during aging can result in the contracting of certain diseases. And maybe a way to understand that is to look at cell cycle genes like this P16 Inc4A protein, right? The result of the expression of that protein relative to cancers. And not only use as a biomarker, then be able to connect the methylation pattern of that gene, because once that gene's methylated, remember it's knocked out of commission, not expressed because of control over RNA polymerase II, the transcription of it. Is that going to link to any alteration in senescence, or is that going to slow down senescence and also at the same time concomitantly decrease the occurrence of some of these diseases, which are going to cause a lot of morbidity and maybe premature, or not necessarily premature, but regular mortality given age. DNA methylation-derived epigenetic clocks therefore perform better than there's claiming transcriptomic and proteomic data. You notice I didn't say lipo data, so not the lipidome, right, but the transcriptome and the proteome. Now, why am I saying that? Because lipids are not directly, at least, related to what? Gene expression. So lipid turnover has a lot to do with transferases, right, and oxidation patterns, and moving groups from one lipid moiety to another, from a sphingosine base to a glycerol base. We're talking about bioenergetics versus single transduction cascades versus membrane fluidity. All of that doesn't necessarily require nascent gene transcription or any of the other things going on in the nucleus, like recombination repair, right, and the chromatin remodeling itself. So you can talk about transcriptomic data, proteomic data, maybe being served by getting a DNA methylation clock generated in the elderly. But that won't necessarily be something you can look at for lipid modification. Right? Now, individual variation like age acceleration, which is defined as the difference between this epigenetically measured age with this DNA clock and the actual chronological age, could be associated with an earlier mortality. That's what they're trying to get at here. So, you go into the doctor's office, you get a blood draw, you get some tissue samples, some other body fluids taken. They maybe one day will do a death DNA methylation clock panel on you, and they'll say, wow, this DNA methylation clock says that you're 75 years old, and the person sitting in front of you is 58, right? So you might tell them that because based on this DNA methylation clock, you have a methylation pattern which puts you much older, which puts you possibly more susceptible to this constellation of diseases that people pick up when they do get in more advanced ages. Now that's the hypothesis, right? That's all that is, a clinical hypothesis at that. But it could be useful if it turns out that what's found experimentally in the research lab does eventually play out in the clinic, right? So it's just the kind of thing you might see as you age. So I'm going to stop here because that was the last slide. It's a beautiful picture of the Colorado Rockies in the fall with aspen trees and some maples. I'm going to say to you, happy Thanksgiving. I hope you have it with someone that you're in your family that you love and you cherish, and that you have a really warm Thanksgiving, and that you appreciate all the people that are around you, your loved ones. Be safe, of course, but also enjoy their company, because no matter what, we're all aging, right? And with age comes the ultimate end of life for some people who are advanced age, and we have to cherish and honor the time we have with them. So with that, I'm going to say bye for now. As soon as I pick up my ability to stop this pathway here.